Welcome, everyone, to the Mowat-Wilson Syndrome Foundation Research Update. Thank you for joining us today. We're very excited. We've had over 150 registrants for this webinar, so we're excited to have that kind of opportunity. Our agenda today will focus on providing an update on current research projects. We've got Doctors Schreiber, Dr. Chatterjee, and Dr. White, who, if you were at our conference last year, all of them presented there. Dr. Vivian will be our moderator to go through a little bit of information concerning cell and gene therapy and talk a little bit about the future of research. But before I turn things over and introduce Dr. Vivian, I wanted to share some exciting news that our foundation has recently gained membership to Combined Brain. Combined Brain is a consortium of patient advocacy groups and foundations that allow all of us to work together, to collaborate, and to develop therapies and share all the work that in the past has been done individually. So just to give you an idea of some of the benefits that we will be able to gain from being a member, one is that they have a biorepository and the ability to collect samples. And one of the things that's very exciting is that we're looking at having a sample collection at our conference next year. And this would be something that's very difficult for us to do on our own. Another opportunity that we have is they have a program called Rent a Brain. I was joking earlier that I wish it was something like that when I was in college, but Rent a Brain gives us access to scientists, to researchers that can help us develop our pathway for making the most out of our process and trying to develop therapies for Mowat-Wilson syndrome. Another example where we can benefit is their registry is Matrix, which, as all of you know, that's what we use. And so by being on the same registry, we will be able to use existing surveys that they have that have already gone through IRB approval. So these are things that are really going to speed up the process for us and give us lots of new opportunities over the next couple of months. The foundation, along with the medical advisory board, will be meeting to discuss how do we take advantage of this great opportunity. So you'll be hearing a lot more about Combined Brain. We will be sharing information with you on social media and updating you to things that we're doing and capitalizing on this great opportunity. As of right now, there are over 90 members, as I said, foundations and patient advocacy groups that are part of this organization. So something that we're very excited about. So to get us started with our program this evening, I'd like to introduce Dr. Vivian. I think most of you know Dr. Vivian, but he is the number one, he's the vice chair of our medical advisory board. He's a faculty researcher, Division of Clinical Studies, Department of Pediatrics at Children's Mercy in Kansas City. He's an associate professor at the University of Missouri, Kansas City. And Jay has just been a tremendous individual for us on our medical advisory board. I believe Jay's been on board from the very beginning, that first meeting in Kansas City in 2017. And he is a great resource for me. He gets my calls. He always is there to help. So it's a pleasure to introduce Dr. Vivian this evening.
Great. Well, thank you, Alan. Thank you, everybody. My slides look okay. Great. Thank you. So now I'm really excited to have be part of this webinar, and we're going to hear some really exciting research from some of our Mowat-Wilson syndrome investigators that we've supported the past few years. But I was asked to kind of give you a little info on a few different things that we want to talk about today. talk about both some of our research, recently supported studies, but I was also asked to give a little bit of perspective on some ideas as we think about heading towards therapies. So ultimately, we want to be able to understand Mowat-Wilson syndrome better with the idea of, can we identify ways in which we can help out our kids? And there are a lot of different options with this, but I think we need to think about what is the foundational work we need to do to allow that research to proceed. And we have a great group of researchers that we're supporting that are going to be central to that. And you'll hear about that. So when we start thinking about, moving towards ideas of therapies, I always like to think about the things we know and the things that we don't know. And, we do know a fair amount about Mowat-Wilson syndrome. We know what the gene is, what is defective, and at a high level, we do understand the basic clinical features. And this table is actually from a paper we'll talk about on the next slide from Dr. Merlin Butler, also in our medical advisory committee, our medical advisory board. And this just highlights the range of challenges that we face across the body of that children and individuals with Mowat-Wilson syndrome have to face. And this is something everyone's very familiar with. So we know Mowat-Wilson syndrome is a complex disease, but there's a lot of unknowns yet, in particularly the molecular and the cellular underpinnings of many aspects of this disorder really are understudied. And we need to know that because that will be the foundation of moving some of our research interest into therapies. And so before I get into that, I did want to mention a recent paper. It was just published by Dr. Butler, a member of our medical advisory board. And this is a nice example of how we could take, our interaction with our families and members of the Mowat-Wilson syndrome, community and move research forward. So in this recently published paper, it was just out, I think in the past few weeks, Dr. Butler's really did a survey of in total of 191 Mowat-Wilson syndrome individuals, and looking at their the genetic defect or the change that's present in ZEB2, and trying to correlate the nature of the genetic alteration with the type of clinical features. And so this has been an interesting study, taking a broad survey of a large number of individuals, and 191 is a large number of Mowat-Wilson syndrome individuals to be able to do this study. And so they're continuing this study to really further understand and try to identify correlations, what we call genotype-phenotype relationships. So, what part of the gene is defective and how that could influence the clinical features. So this is an example of some more clinical research that we've supported in the foundation. But as we move towards therapeutics, we can kind of think about a lot of different, well, what are therapies? Well, we can think of them classically as, drugs or small molecules. These are small organic compounds. Biologics have turned into something that's very interesting. These are usually proteins that can be, applied to an individual to treat certain symptoms. And I want to briefly touch on what I am calling advanced molecular therapies. These are things like gene therapy, antisense oligonucleotides, genome editing, and then also cell-based therapies. These are these amazing front end, almost what we think of as science fiction, type of therapies that are really becoming very feasible and being applied to rare disease. But we also understand a rare disease, which Mowat-Wilson syndrome is, presents its own challenges for any type of therapeutic development. And so, the good news is there's a large body of knowledge acknowledging the challenges of rare disease therapeutic development and how we're attempting to address this. And this paper is actually written by the director of the National Center for Advancing Translational Studies, a component of the NIH, and they've really done a really good job to sort of consider a lot of these different challenges that we'll have to face, and we'll face them together as a rare disease community. So briefly, I just want to touch upon, a couple of ideas that we've been thinking about related to therapeutics. So when we think about small molecules or drugs, these are the classical organic compounds that, would be in a pill or an injectable that we could use as a possible therapy. And so can we identify, a small molecule or a drug that could be useful for treating certain aspects of Mowat-Wilson syndrome? And the answer is probably. And it's a matter of really identifying, what part of the disorder we want to target, what pathway we want to target, what part of the body we want to target, and knowing the complexities of Wilson syndrome will present challenges, but these are things that we'll have to tackle piece by piece. There's a lot of opportunities with using small molecules. A very popular thing that we're doing with some other disorders is repurposing. So in other words, taking an already FDA approved drug and using it in a different setting and a different application for a different disease. And there's a lot of excitement in repurposing, in part because that can really accelerate getting a drug into the hands of the patients and the families. And this is an example of a drug screen we're doing, which is a repurposing experiment to look for possible therapeutics. But there's also opportunities for new drug discovery. So new chemicals, new compounds that we could use, for treating specific aspects of Mowat-Wilson syndrome. When we talk about these advanced molecular therapies, we think about gene therapies. So gene therapies are being applied for an increasingly larger number of diseases. And so what this acknowledges that many of these diseases are defects, most diseases, I believe, are defects in a gene. And so in other words, in the case of Mowat-Wilson syndrome, it's a defect in a gene called ZEB2. And so could we add back that gene through some method and could that be useful as a therapy? And for many diseases, this is finally coming to fruition for gene therapeutics. And right now, adeno-associated viruses, or AAVs, are being packaged which contain the gene that we want to deliver to the patients, and they're applied through a lot of different potential modalities, injection, ex vivo, and things like that. The other category that I'll talk about today are antisense oligonucleotides. And these have gotten a lot of interest in the past few years because they've turned out to be quite successful for a range of neurological, neurodegenerative diseases. And so what this is a sort of a tailor-made compound that can be generated, which specifically targets a gene, in particular the gene product of the RNA that's being produced. And so with the right design and the right target, you can modulate that gene's activity, either turn it on or off, or modify its expression in some way. And that's turned out to be quite effective in some diseases. And I think these are both of these are something that we are looking into and thinking about for Mowat-Wilson syndrome, in part because of some of the nature of the gene that's defective. ZEB2 makes it potentially amenable to these types of therapies. A big challenge for these are the costs for these therapies. These therapies are extraordinarily expensive, and we're working, with the rare disease community to identify ways in which we can make these therapeutics accessible. Finally, I want to briefly mention cell-based therapies. So these are therapies where you are actually introducing cells into an individual's body. The idea being is either the cells in their body are not there, or in the wrong place, or they're defective. And then we're adding them back in. And so this is becoming more and more, many clinical trials and approved therapies for using this. And so a lot of challenges with this, particularly for a rare disease such as Mowat-Wilson syndrome, is because of the range of the complexity of the disease means the type of cell we would want to put back into the body would be very different if we're treating the neurological aspects, for example, compared to, say, the Hirschsprungs or the gastrointestinal challenges. And so, but there is, knowing some of the likely or certain deficits that are present in Mowat-Wilson syndrome, cell-based therapies could turn into something very useful. And we have researchers doing stem cell studies that have the potential to be applied towards that. So, it's impossible for us to talk about all types of therapies, but just want to give you guys a little taste of some of the things that we're thinking about as a foundation and as a medical advisory board. And so, as we move these ideas forward to support research in the future, we always think of the strategies. We need a basis of knowledge, a foundation of knowledge, of a better understanding of Mowat-Wilson syndrome. And that is the foundational work from which, this beautiful skyscraper of a therapeutic can be developed. And we need a Mowat-Wilson syndrome focus, but other diseases, and their experience, and their challenges can be very valuable. And this is where Al talked about the combined brain membership, learning from some of our other rare disease community and taking advantage of the combined brains expertise will be extraordinarily important. And so, ultimately, though, there are gaps in our knowledge, and this is where some of the research we'll hear about today in animal and cell models are really attempting to bridge that. And we talked about the patient data. We'll hear about this. I talked about Dr. Butler's data, and we always think about also what can the foundation and the families do. And we have the registry, the natural history studies, and the community are going to be absolutely central to this, because we need to understand what are the major focus that we need to apply for these therapeutics, and we need participants, an engaged community for this. So I hope this gives us some idea that we could kind of think about, maybe do some follow-up discussions on, and hopefully sets the stage for some of these other researchers that we'll hear about right now. So, we're going to hear now from several researchers, three from our Mowat-Wilson Syndrome Foundation supported research. They're taking diverse strategies. I think you'll really appreciate the diversity of the science that we're going to hear about today. And these are absolutely foundational studies that will increase our knowledge of Mowat-Wilson syndrome, and will absolutely have an impact in how we can address therapies. So I'll go ahead and stop there, and I will introduce Dr. Schreiber, and he will be giving the first presentation. Dr. Schreiber, let me pull up the information here. Dr. Schreiber is a child neurologist and a neurophysiologist at Children's National Health System and pediatric specialist of Virginia. And so, Dr. Schreiber, I'll go ahead and hand this over to you, and real excited to hear about your recent developments in seizures.
Okay, thank you so much, and hopefully you can see my screen.
Yeah, looks great.
Great. Okay, perfect. So thank you so much for the introduction, Jay, and it's exciting to see everything going on in Mowat-Wilson, especially the partnership with Combined Brain. As Jay said, I'm at Children's National in DC. I do work in EEG and epilepsy, and work in epilepsy clinical trials as well. I specialize in other epilepsy genetics. So I'll talk some about the features of seizures in Mowat-Wilson syndrome, talk about EEG and developmental findings, what we know and what we're looking for, and talk about any associations between EEG findings. Some of you may know there is a particular finding in Mowat-Wilson syndrome, which is kind of unique. It's called electrical status epilepticus of sleep. And I'll talk about why that is important, or may be important, and how that relates to seizures and developmental outcomes. So we know that seizures are present in the majority of people with Mowat-Wilson syndrome, and 1 in 4 can have drug-resistant epilepsy. Interestingly, seizures don't seem to correlate with brain malformations. You might expect that to be the case, but not so. It may have something else to do with networks or the underlying pathophysiology in other ways. Seizures often begin in early childhood or late infancy, typically with fever-induced seizures, often focal, that may or may not progress to bilateral convulsions, and later often presenting more with atypical absence that can just be staring spells. And this is something I'll often review with families, is that the appearance of seizures can change over time. So we've talked already about, Jay's very nicely sort of talked about the underlying pathophysiology in Mowat-Wilson syndrome, and there's probably multiple different mechanisms. This is some of the early literature on seizure findings in Mowat-Wilson. So again, focal seizures typically with absence seizures later on. The average age of seizure onset is about 14.5 months, and the majority have their first seizure triggered by fever. So many of you might have gone through that experience of having a quote-unquote febrile seizure, which is not wrong necessarily, only to be later diagnosed with epilepsy, although we do tend to stray away from the term febrile seizure if we think there is some other underlying etiology. Febrile seizures typically reserved for a kid in whom we don't think there is another cause besides the fever necessarily. EEG is typically normal or may show some mild background slowing at the onset, and then after a few years to early school age, we can see diffuse irregular high amplitude spike wave discharges. And if you record sleep, it's really important to record sleep during this EEG. You can see this finding of ESIS, or electrical status epilepticus asleep, and I'll show you what that looks like. So this is what information that came out from the Wilson database. Again, very similar to what had been reported before, maybe a little bit later onset of seizures, but again, fever-triggered seizures are very common, and about a quarter, again, that number, about a quarter are resistant to anti-seizure medications or have drug-resistant epilepsy. If you look at preferred medications, this is what is reported in the literature. So valproic acid is generally fairly effective and used very commonly. We find this for a lot of different genetic epilepsies. Valproic acid is a very popular one and often on that list, in part because it's been around for a long time. If you look at more recent studies, as with ours, you do find there may be some other medications that are used more commonly. So I'll get into that in a minute. I'll skip through some of this, but these are some EEG findings. You can see there's very frequent abnormal activity here on this EEG. This particular patient got treatment with a corticosteroid, which can be a treatment for electrical status epilepticus asleep. And that was really our question, right? Is can we do we see this, and is this associated with developmental changes? And then is it something we might consider treating? So ESIS itself is actually uncommon. It's only in about 0.2% of people with epilepsy. It is classically defined as a spike wave index of at least 85% during sleep. There's a more liberal definition down to 50%. And if you see a significant enhancement of epileptiform activity during sleep, and you see a change in a child's development or regression, then you're inclined to call that ESIS and inclined to treat it. So it can be associated with cognitive impairment, behavioral deficits, including attention and language problems that can persist if left untreated. People often do outgrow it after around 10, 11, 12 years old. Although I think with Mo Wilson, it can stick around for a bit longer. So again, talked about this. This is a patient of ours who has a good-looking EEG during the awake state, looks essentially normal. And then when you fall asleep, you can see this spike wave activity, which in this case probably has a spike wave index. If you look at each 1 second bin, I'd say on this page, probably 60 or 70%, if not more, contain a spike, and that's how we calculate that spike wave index. So recent study in 2021 looked at this a little more closely, and again found spike wave index was elevated at around age 5 years is when you first start to see that. So our objective was to try to understand that not only do we see this and how often do we see ESIS in patients with Wilson syndrome and when does it appear, but we wanted to examine developmental outcomes and see if we found any difference in development and cognitive outcomes in association with ESIS, and also look at other associations with seizures. So here are the patients people we've currently enrolled. Some of you on the call may have participated in this. We have a total of 28 enrolled and are still enrolling. I'll talk to you more about our current plans and what we're doing now. Average age enrollment was 7 years old, so we tend to be kind of young. 43% were male. Very common occurrence of febrile seizures at the onset. So 17 out of 28 had febrile seizures. Most of our patients, these are patients who just had EEG, an EEG capturing sleep with Wilson syndrome. So obviously we are, since we only included patients who had EEGs, our cohort is going to be enriched for patients who have had seizures. So not surprisingly, we had a very high proportion who had a history of seizures. 93%, in fact, had a history of seizures. And the average age of onset was 23 months. And you can see patients have multiple different types of seizures in many cases, and that can increase as kids get older. 8 out of those, the 27 in whom we had EEG results, so about a third of those people had ESIS. Again, recognizing that many patients were younger and may not have yet had the opportunity to develop ESIS. So here are the genetic testing results. I won't spend too much time on this, not really relevant to the discussion. But seizure types, the initial seizure types, again, recapitulating that prior literature shows that focal onset seizures were most commonly the first type of seizure, but there were some other seizure types sprinkled in there as well. Rare spasms, which again, that doesn't happen that often in Wilson syndrome, but you can have epileptic spasms or infantile spasms as the initial seizure type in some patients. Over time, we see that, yes, we do have different types of seizures, including many generalized tonic-clonic and absence or atypical absence seizures that present later on, and some patients have multiple seizure types. Anti-seizure medications, I alluded to this before, but you see that levetiracetam was used most commonly. There was a good response, so that usually means at least 50% or more improvement in seizures. In most, a poor response in some, and not otherwise specified in a small proportion. But our most effective medications and most commonly used medications we found were levetiracetam, followed by clobazam, oxcarbazepine, which will probably be used more for focal seizures, valproic acid, and then some others. So we looked at the risk of developing ESUS with age, and we found that around 60 months would be 5 years old. That's when you start to do see a bit of an uptick in that ESUS incidence. Note that this includes the proportion diagnosed with ESUS at the age of EEG when they were first diagnosed with ESUS. So some patients may not have had an EEG exactly when they developed it. I think some patients are much older, and EEG may not have been necessarily used right away for some of those patients. But a pretty high proportion as they get older did develop ESUS. So we looked at developmental milestones. We did try to get a sense for when people acquired developmental milestones and what they did. So we picked out some more of the salient milestones. This is a bit of a busy slide, but this is from the CDC developmental milestones that are published. And looking at language, we find that most patients are able to coo, issue syllables, laughs, and may say single words, but many can't combine words or certainly speak in full sentences or write. In the motor domain, many can reach, grasp, and scribble, but can't draw a circle or draw other more complex things. Most have head control and can walk upstairs. Some can throw, most cannot hop. And then socially, kids are a little better. So these kids actually do pretty well socially. Many of you know that already, but they're very happy, smiles. Some even help with chores and can help with dressing and that kind of stuff. So if you look at the Vineland's another way to look at development. We find that, again, the most advanced skills, the higher the score, the better your development. And these are age adjusted. Were found for socialization, and the worst, not surprisingly, is for communication. And many of our patients are using augmentative and alternative communication devices as well. If you look at the adaptive behavior composite, which is kind of everything combined together, it looks like these things decrease a little bit as patients age. What's probably happening here, though, realistically, again, we only have single points in time, but what's actually probably happening is patients are just falling further behind their age-matched peers. I'll skip through some of this. This just goes to the subdomains. We did not find any, just to jump to the conclusions here, did not find any difference on the Vineland in ESUS versus no ESUS, although I think you do see that the scores are lower for those folks that have ESUS. Because there was a difference in age for patients with ESUS versus without ESUS, we did adjust for age and did not see any difference after adjusting for age on those Vineland scores. We picked the Vineland because it's used for a lot of clinical trials now for other developmental and epileptic encephalopathies or genetic epilepsies. But I think we're finding that it may not be so well suited for this population and other populations. So that kind of leads to some of our next plans. So here was age for those diagnosed with ESUS in years, and the current age in those who did not have ESUS. Again, I think you see that there may be some in here who do develop ESUS that were not noted to not have ESUS at the time of inclusion. We did find that the trend toward significance in patients who had intractable seizures were more likely to have ESUS than those without. But notice again, our numbers are fairly low. So we also asked about regression of development. Didn't find any difference there. and no difference in patients who had regression with or without intractable seizures. So future directions, we are now changing our IRB so we can include some different developmental measures, which I think are actually going to be much better suited to this population. We are in process of doing that right now, so I'll give you our emails if you're interested in participating in this and it did not before, then please let me know, because we really want to expand this cohort and roll out these developmental tests soon. Expand data collection, look more at EEG and do more EEG analysis. We talked before about incorporating some survey questions into the registry too, just to understand more about seizures and ESUS. So here are the references. I just want to thank our team, and particularly the Mowat-Wilson Syndrome Foundation and all of you who've funded this and have really contributed so much to this work, just by providing your stories and everything too. So thank you so much for your time. Here's my email address and my research coordinator's email address that is working on this. If you'd like to participate or have any questions, please, please, please do not hesitate to email, reach out. Thank you so much.
Thank you, Dr. Schreiber. We got a minute or so. There was a question in the chat talking about illness-induced seizures. So Doreen talked about a virus-induced seizures. Can you comment at all about how, illness-associated seizures are maybe, that Mowat-Wilson syndrome may be more preponderant in those situations, or does your study attempt to address that?
Yeah, so we looked at it not necessarily with illness, but with fever. So fever-induced seizures, again, are something we found very commonly in patients with Mowat-Wilson syndrome, particularly early on. So we consider febrile seizures to occur between 6 months and 6 years old for the general population. This is not just for Mowat-Wilson, but for anybody, because we know that kids at that age are particularly susceptible to seizures associated with fever. We didn't look at any particular type of illness or anything, or really at fever-induced seizures beyond that point. But I think that is some, an area of study that probably warrants additional exploration.
Great. And how much of a larger cohort would you like to see for your study to really power it up and make it really, if you had your druthers, what size study would you like to see for this?
Yeah, I think to detect, I don't have those numbers at my fingertips right now, but when calculating this initially to detect what we would think to be a clinically significant difference in developmental measures and to have a sufficient number with ESUS, we like to see more like 50 or 60 total. I think more is always better.
Absolutely. Well, it's a great opportunity for our foundation to hopefully encourage some participation and move forward.
Thank you so much.
Great. Thank you, Dr.
I apologize. I have to hop off for another meeting.
Awesome. Great. Appreciate your time. So,
and I'll stop sharing too, so you can share.
Yeah, that'd be great. All right. So our next speaker is Dr. Sumanta Chatterjee. So Dr. Chatterjee, feel free to load up your slides now. Dr. Chatterjee is a research assistant professor at NYU Grossman's School of Medicine in the Center for Human Genetics and Genomics. And so we're going to hear some really interesting and exciting models that he's doing to study the enteric neurons in Mowat-Wilson syndrome. So please go ahead, Sumanta.
Right. So you can hear me, right?
Yep.
Okay, great. Well, I was muted. So first of all, thank you, the foundation, for giving this opportunity. I've always been absolutely awestruck by the work you guys are doing and the families who contribute to all these diseases. So I'll sort of change focus a little bit from Dr. Schreiber talk about. I am not a clinician. I'm a researcher. My interest has been on the enteric nervous system, and my entry into Mowat-Wilson was via the GI tract or Hirschsprung disease. But before I jump into those, there might be people in the audience here. Oh, also I've given my email here. So if anybody's interested in contacting, please feel free to, after any questions I don't answer, I'm happy to help. I thought I would give a little bit of a background of who am I and why do you guys have to listen to me? So I was trained as a developmental biologist studying mouse genetics, looking at bone and cartilage development, and there was no disease focus in my life in those days. But when I trained as a postdoctoral fellow at Johns Hopkins here in the US, I sort of got into the world of human genetics and diseases, particularly congenital or childhood disorders, and among them, primarily Hirschsprung disease, which many of you in the audience are really aware of. It affects children, it affects the GI tract, it affects the enteric nervous system. It's left untreated, it's lethal. Now there's surgery for it. But so that sort of piqued my interest in the cell system and its associated disorder. So independently, as I moved on in my career, I've sort of focused on this system and its diseases. As you know, Hirschsprung is the primary disorder of that class, where kids are born with almost pretty much absent or reduced neurons. They can't pass the stool. Right now, the only option is surgery. If they're diagnosed, they surgically remove the portion of the GI tract which has no neurons. But I got into Mort-Wilson syndrome because 50% cases in Mort-Wilson syndrome has Hirschsprung disease, and which to me is still surprising given that it's a single gene disorder. Why doesn't everybody get? Which suggests to me one part of the project I was doing with the foundation is sequencing individuals to figure out if in some of the individuals who do get Mort-Wilson has more than one mutations beyond ZEP2. Because logic will dictate if you have a single mutation, everybody should get the same phenotype in general. But that's another story. But so that's my entry into it. I was very interested and intrigued, and the foundation was kind enough to allow me to sort of get in there, talk to people, figure out what is going on, and sort of take this aspect of research into my research laboratory to figure out at least the GI or the gastrointestinal problem of Mort-Wilson syndrome, and that's where the focus has been. So a couple of questions we're broadly asking, which covers the diseases we work on. One is, as you know, from any of you who have done the sequencing nowadays, with the advance of sequencing, we're getting many, many mutations in every patient. So, and we sort of have a catalog of this. Yet there's a poor understanding of why certain mutations cause certain phenotype in the end. So the lab is trying to figure out experimentally why a particular mutation causes X and another mutation will cause Y, and if severity of a disease is association with the type of mutation. It's an academic question, of course, but there's also a larger patient classification here, where if you have a large catalog of what you call genotype to phenotype correlation, every sequencing screen should be able to predict what the outcome of the mutation would be. So that's one aspect. But as Jay alluded to in the beginning of like going forward, there's a cell therapy potential. So we also want to figure out if the specific mutation leads to a very specific cellular phenotype, because if you want to do cell therapy, you better know what cell types are you're going to therapize on. You just don't go in there and try to change every cell out there if the cells are not effective. And then from a perspective of this is, as you're very well aware, it's a developmental disorder. So you're already born with it. So if you also want to therapy, you will know what kind of processes these mutations affect, what exactly is going wrong in these cells when you finally see a mutation, what did the mutation do in the process? Again, there's large implication in therapy, because if you want to target something, it's better to go in there with as much knowledge as you can. So one thing we have done over the last decade or so, we have built a very nice network of, so one first step towards doing any of this is understanding what genes are involved. So like in Mort-Wilson, we know ZEP2, but in Hirschsprung disease, there are multiple other genes involved. And sort of ZEP2, we now know feeds into this network. So because if you want to do gene testing, instead of doing the whole genome, we can now do a very targeted sequencing of patients and ask the question, do we find mutations in any of these genes? Now we know the genes are interconnected, they control each other. So you have mutation in one, you can predict the outcome of the complete disease. So it sort of gives us a much more principled way of looking at patients now when we get DNA samples. Instead of like going gung-ho into the genome, we have a much more specific questions. The second thing which sort of gets me to the top of Mort-Wilson is if you do any sort of experimental purposes, we always are stuck with two things. One is, what do we test for and what do we test it in? Because you cannot go test it in human beings. So you're going to need an assay and you need a cell type to test it in. Now in Hirschsprung and even Mort-Wilson cases, it's a defect from our studies and others it's a defect of proliferation. These neurons don't migrate to the end of the gut. So the end of the gut has no neurons. So if any mutation you create, the thing you can measure if this mutation leads to this, what you call migration defects, and these cells can't divide. So does it lead to a proliferation defect? So any test of function has to measure these two. And I will show an example how we have done this. And then we need a cell type to test it in. So you cannot sort of go in there and test in a kidney cell for a disease of the gut. So now we have developed two cell types. One is it's a very classical neural crest derived, because these ENS comes from a very single cell type. So we have derived a cell type which sort of mimics the ENS, and we can test rapidly in those cells, any mutation you give us. But the real test is, can you do it in a ENS, the actual nervous system derived from the patients? And so we have now started deriving ENS from patient-derived iPSCs and skin fibroblast. So which basically now we are not touching the cells at all, except deriving them. So the mutations are exactly what the patient had. Every other change is in the patient. So it's a physiological system. We have not disrupted in any way by ourselves in it. So this is now a workflow which we have sort of over the last couple of years, many people have worked, which we can do it when you don't have patient cells and you give me a mutation. I can take a cell, which I told you about, the neural crest cell, and I can edit that mutation in, and then I can rapidly grow these cells in plates, and then I can measure the two assays I talked about. One is proliferation. Can these cells sort of divide and grow and become more in number? And can they move? Because that's what the defect is, the absence of this movement. And because it's quantitative, we can measure amounts. You can actually now classify if mutation A leads to a stronger phenotype than mutation B. And this is a measurable assay now. So that was one of the bottlenecks in our field. And we're sort of working rapidly towards now getting this out of the system. So this is not all cartoons. We have now done this. This is only in patients with Hirschsprung disease. Actually, there's one patient, I think he had Mort Wilson. So we've taken specific mutations in patients on a single gene called RET, and we have now created those mutations. So each of this letter tells you one amino acid as in this patient to transfer to another amino acid in that patient. So originally it was in lysine, it became methionine. It was a tyrosine, it became a phenylalanine. And we can now measure the change of this proliferation. This is basically how fast the cells are dividing over time. So from 0 day to about 0 hours to 100 hours, how quickly the cells divide. As you can see, some of these mutations has no effect. These mutations are almost like a wild type cell, which basically means that the patient carries this mutation, that's not the causal mutation. Whereas some of these mutations, where a tyrosine becomes a stop codon, basically the truncation, division of the cells completely almost goes to zero. The cells die over time, which basically means they are more causal than these, completely causal. Similarly, the assay is migration. Can the cells sort of migrate or move? Because if there's mutation, again, you can quantify this. The pictures, the top, you can show basically we plate the cells and we draw a line between the cells and divide them. And over time, you measure how quickly can they cover this gap, right? This is also called a gap closure assay. And each of these, again, at the bottom is an exact mutation we created in the cells. So E became a Q, a tyrosine became a stop codon. And now you can measure how much of the gap is actually closed over 3 days. So you can see at the top here, this cell here pretty much completely closes the gap. So if you have the specific mutation, E178Q, you don't have that much of a defect. I'm sorry, this is for the wild type. But if you have some of this kind of a mutation, F998L, the gap close is much lesser. So it basically means if you carry that specific mutation, those cells cannot divide, but they can't also migrate very well. So this sort of gives us a foundational basis of measuring mutations. If you give a mutation, we can now start looking at its effect in this thing. So we sort of thought we'll take this. Oh, sorry. So finally, we can now start, once patients gives us, then there was a prediction from software which tells us what these mutations were. We can now do an actual assay and tell you what was predicted to be benign is actually benign, or what was predicted to be an uncertain significant. We can now call them either a benign or a pathogenic. And in some cases, what was predicted to be pathogenic is pathogenic. So we can start reclassifying variants, what was predicted, and are truly functional if those variants are variants. We're in the process of scaling this up. You can give us any type of mutations we're trying to do specific Mort Wilson ZEP2 mutations to see if we can sort of rapidly spin it up. But for Mort Wilson, Dr. David Mort gave us two families, DNA from two families and a little bit of fibroblasts in both the families. In one family, the child had Mort Wilson and Hirschsprung disease, and in the other family, the child only had Mort Wilson, no Hirschsprung disease. So there's a family where there was a kid who had Mort Wilson and long segment Hirschsprung disease. And they told us the mutation, what it was, but we did a whole genome sequencing just to reconfirm the mutation, as well as to see if there's any other changes. And we also sequenced the two parents. And so the parents had no mutation, of course, any other, and the kid, of course, had this known mutation, and we didn't detect any other pathogenic variant in this child. So we derived now that I told you before, those cell lines we initially worked on were not really ENS. They are neural crest cells, which we made mutations in. But now from these patients, we can now take the fibroblasts and derive something called induced pluripotent cells. These cells are cells which are converted back to a state where they can form other things. And we sort of made them entry neurons, we derived. This protocol is slightly newish. 3 or 4 labs in the world are doing it. We are one of them. And so this yellow color marker, TUJ1, is basically tells you these cells, once they start from ENS, they have become neural cells. They've started expressing markers which are known neuronal. So these cells are actually in a dish, they're behaving as entry neurons. So, which was great. So which sort of tells you the protocol, though it's still a little right now not very stable, but it can be done. It sort of takes some time and some tender loving care, but you can do this. But our real question for all this was, if you take this, as again Jay alluded to, you want to do cell therapy one day, the field, we need to see if there's a defect in the child, what is actual cellular defect. So we can take these cells in a dish, we can dissociate them, and we can sequence these cells. And by sequencing these cells, so each dot here, colored dot here, is a single cell from that dish, either from the father, the mother, or the child who had the Mort-Wilson syndrome. And we have measured gene expression or changes in gene expression in these cells. And we can count these cells now, what type of cells they are. As you can see in this patient, so the orange here is the child with the Mort-Wilson. It's a nerve cell, the neurons, which are mostly defective. Every other cell type is pretty much normal in this derivation. And then we can take these neurons and we ask what class of neurons are effective. So we can, based on what kind of genes they're expressing, we can classify them as nitrogen neuron, acetylcholinergic neurons. Some of these you might be aware of. Acetylcholinergic neurons release a hormone called ACHE, which is a standard stain in the clinic for Hirschsprung patients. That stain basically stains cholinergic neurons. And we can show in the one patient we had from whom we have derived this, we can see multiple clusters of neurons are affected. It's not a single neuronal disorder. Now, this n is equal to 1. This is we're doing it the first time. So there's caveats to this, but at least we can sort of have this protocol in place. We can count these cells and we can see the mother and the father are absolutely normal, which basically suggests that the process is working. Our cells assays are working. Now, this is unique to this derivation is unique to the cells. We're still in the process of sort of optimizing this, but at least the first forays into understanding is a Hirschsprung disease in a Mort-Wilson case, what kind of neurons are missing in the child. We're doing it also in parallel with just Hirschsprung cases who doesn't have Mort-Wilson to see if the Hirschsprung in a Mort-Wilson versus Hirschsprung in an isolated Hirschsprung case of how similar the ENS looks like. The other thing I told you, we can measure proliferation. So the idea is, are these cells also defective? So we can take the same cells I showed you. And again, this is basically an assay to how quickly these cells divide over time and how quickly the cells migrate. So the red and the green dots are from the father and the mother. So over time, these cells sort of divide very rapidly in this individuals, the father and the mother, and they reach a confluence in a dish. Whereas a proband who has a disease initially starts dividing and the cells then stop over time. And over days, the cells can no longer divide because of the mutation. It can't divide anymore, and slowly the cell starts dying off. Similarly, the cells over time can't close the assay. So the cells still migrate. It's not that they don't migrate at all. But as you can see, the father and the mother, they pretty much migrate much rapidly, whereas in the proband, the child, the migration is much more deficient. Slightly linked because the fewer cells, remember, the cells are not dividing here. So the cells are, because they're not dividing and proliferating, they also the migration slow. So these are two linked processes. Because they are not enough of them, they can't go further. So it's a critical mass issue. But we can do this also now. Again, we've done this a couple of times in a way, sort of setting up a system which we can sort of, going forward, we can do this more rapidly. So I will sort of stop here, but as told you, so as Jay alluded, this is one of the methods where people will go on and slowly start looking at these processes. So we're still in the early days of deriving ENS, unlike many other systems. So there's still a challenge of doing it with a few labs doing it around the world. So we're still learning as we're going along in a way. And so also we have to realize how much of this phenotype we see in a dish is truly what happens in a patient who has the disease. So getting tissues from patients, gut GI tract tissues, and looking at it as sort of one of the things we are looking at both in Hirschsprung as well as associated diseases like Mort-Wilson. And I always end with this, any foundation talk I give. So we have to remember this is just because it is rare, it's not unimportant. So there are lots of kids out there with rare diseases, and any assays, any information, any genetic screens we do will not only affect Mort Wilson, but largest screen of rare diseases. And so the more we come together, the more assays we create, the more methods we generate, it'll be better for everybody. And including getting funding agencies to look at us and say, this is not just a few kids who nobody else looks at. It is not unimportant. So I really again want to thank the foundation for sort of picking up the project, doing this. It is really, really important. None of this work is done alone. I mean, there are many, many people involved in the lab, people in my lab, primarily Lauren, who has now moved on. She did a lot of the early work on setting up these assays. And now Maninder is doing some of this work. Other labs we work with David Moore. David has been very, very helpful in sort of indulging my questions and things whenever I asked him. The foundation for funding us initial studies started with the foundation. Thank you for listening. And I can answer any questions. I'm happy to. I can
Great.
stop sharing, I guess.
Yeah,
There you
that'd be
go.
great. Thank you, Dr. Chatterjee. I think in the interest of time, we'll have to move
Sure.
on. We got a couple questions, and maybe we could have you follow up on
Sure.
offline on
Sure.
that. So that would be great. Thank you. It's very exciting work, and look
Thank
forward
you.
to seeing the progress. Yeah, so we'll move on. Dr. White, if you could load your slides. Dr. Stephanie White is the William Scheibel Term Chair in Neuroscience, and she's professor of integrated biology and physiology at UCLA, and is also the undergraduate neuroscience chair of their interdepartmental program. And so Dr. White will be giving a very exciting presentation on an animal model related to speech. So please go ahead, Dr. White.
Thank you very much. Can everybody see my opening slide? Seems
Looks
okay.
great.
And hear me. Thank you.
Yep.
So I want to thank the board and the community for the opportunity to provide some updates about our work on ZEB2 since the amazing Mowat-Wilson Syndrome Family Conference last summer that I was lucky enough to be able to attend. So as this slide shows, most of you know my lab uses songbirds to try to understand how brains give rise to learned vocalizations like song and speech. And recently, the free resource of the transmitter was launched, and this was by the Simons Foundation for Autism Research and Initiatives. And I really appreciated the title of this topic, which is that non-model organism renaissance has arrived. So you can see a variety of non-model organisms are making their way into neuroscience, and one of them being birds. And I want to thank the Mowat-Wilson Syndrome Board for recognizing this a little bit earlier than the Simons Foundation did, and in giving us some support here. And I don't know if the Simons is involved at all with the combined brain group or not, Al. I don't know if you know about that. But anyways, in my lab, we do study a certain species of songbird, in addition to some others and parrots, and that's known as the zebra finch. And if you were at the conference, I passed around little replicas of these birds. And the reason we focus on them is because they are vocal learners. And we hope that what we understand about how their brains solve the riddle of how to communicate vocally is going to be shared with other organisms, including humans and children. And like humans, zebra finches learn during developmental critical periods, and they learn through social interactions with their siblings and their caregivers. And they need to be able to hear in order to know how they should sound. And despite the fact that humans and songbirds did not have a common evolutionary ancestor on the family tree that had the trait of vocal learning, it appears that the brain has nevertheless found a similar mechanism for supporting it. And so they're going to show you evidence of similar circuitry, and we think similar genetic underpinnings. So just as a reminder, kids start out, babies sort of passive listening. They have their innate vocalizations, their cries. You wouldn't want to have to learn to cry in order to get attention right after you're born. But then a little bit later, babies start babbling. And that's really the beginning of them starting to practice how to control the muscles, orofacial muscles, in order to speak and sound and give their first words. And so similarly, young zebra finches are hatched, and at first they're just listening to others to see how they're supposed to sound. And then there's a certain developed milestone where they also start babbling. And so we call this first period for listening, the sensory acquisition period or critical period, and then the second period where they're both listening and practicing, we call that the sensory motor critical period. Okay, so similar critical periods. What about this idea of similar neural circuitry? Well, what I'm showing you here are two brain sections that are taken in the sagittal orientation. So sort of like this for me, or in the orientation that you can see on the slide from a female and a male zebrafinch. And in this species, but not in all songbird species, but in zebra finches, vocal learning is a sexually dimorphic trait such that the male learns a courtship call in addition to his innate calls, and the female has her innate calls, but she doesn't learn a courtship call. Instead, she evaluates the male's courtship call. And so if you stain the brains of the male and female, as was done here, you can see groups of neurons in the male that don't appear as evident in the adult female zebrafinch. And so that gives rise to the hypothesis maybe these are important for that sexually dimorphic trait of vocal learning. So a lot of research by a lot of songbird researchers has shown there are other parts of the circuit, but this is a schematic, and these are the two cortical regions that I think are most relevant for our audience today. This group of neurons shown up here with the black arrow and circled in red in the schematic called HVC, which is just an acronym that we use as the proper name, and another cortical area down here. And this one is really primary motor cortex dedicated to learned vocalization. So for whatever reason, songbird brains clustered all the neurons working together to learn and produce song into groups that you can see even with the naked eye in a slice of brain. So the male bird has this pre-cortical to primary cortical connection, and then a direct connection onto the motor neurons that produce singing. So one really tantalizing piece of evidence is that during development, we know that this premotor area sends axons down to the primary motor area at a certain point in time, and that's 30 days after hatching. And it actually happens in both male and female brains. And then, for reasons that I can answer later, the axons go in and actually synapse, make connections in the male, whereas they retract in the female. And so that's one of the reasons, the brain reasons why we think it's sexually dimorphic. And when this happens in the male, that's when the onset of the critical period for bird babbling begins. And I'm not sure that that such an anatomical hallmark has yet been identified for when in human babies they start babbling. So we went ahead and since I spoke with you, we started looking at the development of ZEB2 expression in this pre to primary motor circuit area. So just remember up here, we're focusing on HVC. And so we looked at 25-day-old male and female brains. These are Krussel stains, just showing you where in the section that HVC should be. And then this is a DAPI stain and all that's slightly larger magnification, just showing you where cell nuclei are. And then in red, we're staining for ZEB2. And what I hope you can appreciate is that at 25 days in the male and 25 days in the female, there's some signal here, but not a lot. But by the time the male is an adult, there's a lot of ZEB2 expressed in the male and not in the female. And you can appreciate that maybe a little bit better in the merge. So ZEB2 expression becomes enriched in the male, but not the female HVC. And so my graduate student then decided to examine the expression of ZEB2 in the adult male brain and compare it with GFP labeled tracks that are going to label this pathway that goes from the premotor to the primary motor area. So you can see the expression of ZEB2 in the premotor area. And now, thanks to an AAV expressing GFP, you can start to see the tracks going down to the target area, which is the primary motor area. And if I blow that up for you, you can see that this virus, this AAV virus that's just expressing GFP, you can see fibers in this pathway, which is the critical pathway for bird babbling. And to see if this virus is actually getting into neurons that express ZEB2, here is a blow up, and you can see in red ZEB2, which is a transcription factor, is normally in the cell nucleus, and then that some of these viruses are hitting those cells. So we are pretty excited about that. And again, just reminding you that in baby males, there's tons of this ZEB2. Then in adult males, it becomes rather restricted to the HVC region here. And then when we look at adult females who do not have this pathway anymore, we see really low expression. So we used this same AAV, and remember, Dr. Vivian mentioned AAV therapies, also antisense oligotherapies. Those are both things that we're trying. And so we injected an adult female with an AAV that would express ZEB2. And this is very preliminary data, but we think we're seeing in red now signal, and maybe this injection actually, we might have let some virus out in this hyperpaleal area, but this is really where the HVC would be. And so we think we're seeing enriched expression in the female with the ZEB2 injection. We need to do more of that work, and we're doing it right now. But another thing that was very interesting, given Dr. Chatterjee's comments about cell migration, is a feature of the songbird brain is that there's ongoing neurogenesis throughout development, even in these cortical neurons. And so you can mark them with a marker called double cortin. And generally, the neurons start at this ventricle area, and then they migrate out into the cortex. And so you can see them in green. And you can see some of them double label here for ZEB2 and double cortin. And what we're really wondering is if they are proliferative, and then they go to where they're supposed to be, and then they turn down the double cortin and upregulate ZEB2. And this would be a very exciting finding and sort of mirror the potential role of ZEB2 in migration in the central nervous system, similar to possibly in the enteric nervous system. So we are developing oligos, antisense oligos, to do the converse, to not just overexpress ZEB2 in females, but to maybe knock it down in males. And for that, we really want to be able to assess small changes in ZEB2 expression. And so we're very happy with this Western quantitative way to measure. Here's ZEB2 with differences loaded here, two 2 isoforms and a loading control. So we're hopeful that we're going to be able to pick up any small differences in ZEB2 expression as a function of the oligo. And so I'll just end by reminding you that vocal learning is a convergent behavior that evolved in birds, but also in primates. And so we hope that what we learn about ZEB2 in songbirds is going to be applicable and helpful. And our take-home messages are: we're using a comparative approach to identify convergent specializations of cortical brain connections. Could ZEB2 expression initiate the human brain connections for speech? Will knocking it down in males decrystallize adult song or prevent the initial connection and babbling? And will overexpression in female HVC cause that vocal motor backbone to remain and for females to be able to sing? So again, I want to thank my team here that did the work. Greg Gedman was one of the early contacts with Al, and we received the Macy Wisner Research Grant from the foundation. So thank you, and happy to answer any questions now or later.
Great. Thank you, Dr. White. One question, and we'll probably have to move on in the interest of time, but there's a question brought up is, does this mean that Mowat-Wilson syndrome could be an interesting gene therapy target, at least for speech language acquisition? She was under the impression that Mowat-Wilson syndrome may not be a great target for gene therapy, since historical understanding was ZEB2 was crucial to brain development in utero.
I think that knowing what the effects of manipulating it post uterine environment or egg, I think the bird could really help us understand what the likelihood of rescuing something post neonatal neogenesis would be really interesting.
Great. Yeah, I think this is another great example of, as we discover new functions and activities of ZEB2, this opens up the potential for new therapeutic directions. So, okay, well, thank you to all of our speakers. Great topics, great discussion. Al, I'll turn it over to you for wrap up.
Sure. Well, first of all, I want to thank everybody that listened in today. And we'll be posting this on our website, and we'll send the link out so that if you registered and didn't get a chance to participate, or if you did and want to go back and listen to something again, you'll have that opportunity. Again, like Jay, I want to thank our presenters today, Dr. Chatterjee and Dr. White and Dr. Schreiber, and then of course thank Jay for his efforts. But I think you see and are starting to understand that we're at a very exciting time for our community with all that's going on in the world of research in general, and then bringing it down more specifically to what's happening for Mowat-Wilson syndrome with our current researchers. And as I mentioned earlier, I think our membership in Combined Brain is going to open even more doors for us. So the future in terms of research for Mowat-Wilson syndrome is very bright. And I'm excited to be part of that research committee and working with such great researchers and clinicians. And our goal is to keep the community up to date as much as we can with everything that we're doing. So thank you again to everyone.
Thank you.